All right, we're going to go with some virtual learning today. So this recorded lecture is just to help you better understand the content for chapter 23. Some of our announcements coming up. So your quiz, since we're not in class, is through the Kahoot. So you just need to take it like we would in class just sometime today. Be sure to complete it though by 11:59 tonight. Your chapter 23 assignment is still due at the end of the day tomorrow. Our exam 4 is still scheduled for Tuesday on all the chapters that we were planning. Just a reminder about the SEI bonus. Again, you don't need to send me the screenshots for those. Instead, everyone will get 5 points as long as 75% of students complete the assignment, and that is open until the 7th. Actually, that's open. It's a little bit longer than that. It's through the last day of classes, which I think is actually the 12th. So sorry about that. And then our final exam is the Wednesday of finals week, which is the 18th. Just an announcement here that we were asked to share. So there's a workshop for finals. If you want some help about preparing for those. Also have some pizza and refreshments as well for that. That's going to be Tuesday, December 10th from 4:30 to 5:30, and that's in the library. G6 is actually in the bottom of the library, but they'll help direct you there if you get to the library. All right, our learning objectives for chapter 23 are going to be talking about the Hardy-Weinberg principle and how to determine if evolution is occurring within a population. We'll also talk about non-random mating and genotype frequencies. And then some of the things that drive evolution, which is natural selection. There are different types of natural selection, like directional, stabilizing, and disruptive. There's also essentially sexual selection, so inter and intrasexual. And then other driving forces of evolution, genetic drift, gene flow, and mutations. So first of all, just understanding the difference here between individual genetics, which we've already covered. That's our Punnett squares, where we have, our, in this case, heterozygous parents for an allele, and then that individual genetics showing all the possible kind of combinations of our egg and sperm to show possible offspring. And that's for an individual or offspring from a certain person or a mating pair. Here, whenever we're talking about evolution, though, we talk about a gene pool. And a gene pool is all the parents in the population and then all the possible gametes. So again, we're not just talking about two people mating, we're talking about an entire population. So if whatever that population, if we're talking about a city or we're talking about a country, we could be talking about an ocean or a pond. Again, that population is defined by whatever area they're looking at. But essentially here, instead of looking at two people mating, we're looking at all the possible eggs and sperms in that population. So that's what makes up what's called the gene pool. And in this situation, we're having kind of Mendelian genetics, so a dominant and a recessive trait. So in this case, we're seeing the dominant trait occurring about 70%, so 0.7. So if we add them up and divide by the total number, and about 30% for the recessive allele. So then which offspring genotypes are likely? And again, this is based on the population because this is assuming random mating. So it's not pairing up individuals. So any of these individuals could come together. So then what we have is essentially 70% dominant traits, 30% recessive. And then you assume that kind of heterozygous mating, because again, this is talking about the whole gene pool of how likely these are to come together. So then the possible offspring genotypes, whenever we have these two alleles, again, a dominant and a recessive, there's 49% chance of having the homozygous dominant, 42% of having the heterozygous, and 9% being homozygous recessive. And then the next slide, we'll see kind of where those numbers come from. The next slide. All right, so we have what's called the Hardy-Weinberg principle that leads to this equilibrium. And the idea here with the Hardy-Weinberg is that this is assuming no evolution, so that there is an equilibrium or a balance and there's not being a change in the allele frequency. All right, so then we'll talk about how do we know if evolution is happening. So the Hardy-Weinberg equilibrium or principle, again, is reaching that point of 70% to 30%, or just having a continuous frequency over time, so no change over time. Again, here what we're focusing on are two alleles for a gene. So we have A1 and A2, and in this case, if we're talking about true Mendelian genetics, A1 would be dominant, A2 would be recessive. And then these are given symbols. So the frequency of the dominant trait is P, and the frequency of the recessive trait is Q. And again, this is in the gene pool. This is the whole population. So of all the egg and all the sperm that are there in that gene pool, the frequency, the number of dominant alleles to the number of recessive alleles. So because those are the only two options, then we know that the frequency of the dominant trait and the frequency of the recessive trait would equal 1. So if we only have two traits, then they add up to 1. So again, that's 100% of the population. All right, now back to where we get those numbers from, and just kind of explaining this. So again, here's our parental frequency. So again, this is our gene pool. These are the eggs and sperm that are here. So if we count up all the dominant traits, there are 28 out of the 40 have that capital A. 12 out of the 40 have the recessive trait. So again, the dominant trait is P, the recessive trait is Q. We add both of those up to give us 1, and that's what we happen. So there are 28 dominant, 12 recessive. So if we divide that number by the total number, so there are 40. So if we count up all of these A's in here, there are 40 A's. 28 of them have the capital, 12 have the lowercase, so the recessive trait. So 28 divided by 40 gives us 70%. 12 divided by 40 gives us 30%. Again, 0.7 plus 0.3 equals 1. All right, so then if that's our possible gene population, now if we use our Punnett square, now if we look at genotype frequency. So again, understanding the difference here, P and Q are representing the alleles. So each individual gene for that trait. Okay, so P and Q again are the allele frequencies. We can use those to determine the offspring genotype frequencies. And now we're talking about the homozygous dominant, heterozygous, or homozygous recessive. So if we use this Punnett square based on those numbers, what we have is we take the dominant trait, so AA, so capital A, to give us our homozygous dominant. We determine that frequency of the genotype, again, the frequency of having homozygous dominant, and we do the mathematical equation of P squared. So we know that P from our information up here, P is 0.7. So if we take 0.7 and square that, so 0.7 times 0.7, that gives us 0.49. So the genotype frequency of the homozygous dominant trait is 0.49 or 49%. Now the heterozygous, we have one here, so 0.21. So, and we get that by multiplying 0.7 times 0.3 to give us 0.21. So that's P times Q. So the dominant allele times the recessive allele gives us our heterozygous. Well, that happens twice. So we get the heterozygous combination is possible twice here. So we get the frequency of the heterozygous by taking 2 times P times Q. So in this case, 2 times 0.7 times 0.3 gives us 0.42. So there's 42% chance of having the heterozygous. And then finally, we know that the little a from our allele frequency up here is going to be Q. So just like the homozygous dominant, we take we take Q squared. So 0.3 times 0.3 gives us 0.09. So the allele the genotype frequency for the homozygous recessive is 0.09. Now, if you add all of those up, that also gives you 1. So this is called, let me just get rid of a lot of this writing here. All right, so then that gives us this Hardy-Weinberg equation, which is P squared, so the dominant allele frequency squared, plus 2 times the heterozygous, so P times Q, plus the recessive trait, Q squared. Those also add up to 1. Okay, so that is called the Hardy-Weinberg equation. P squared plus 2PQ plus Q2 or Q squared equals 1. That is the Hardy-Weinberg equation. You will be doing some examples coming up here, and you will be doing this on your test. I will give you this equation, but you will need to understand what P and Q are to understand how to answer the questions and to apply this, this equation. But I will give you this Hardy-Weinberg equation. All right, and then finally, what we have down at the bottom is the offspring allele frequency. So again, 49% is the homozygous dominant, 42% is heterozygous, 9% is the homozygous recessive. So if we know those, we can also go backwards. So if we know that the gene pool here of the next generation, okay, so now this was this, so now whenever these offspring, whenever they mate and they have our next generation, then what we can do is determine the contributions for each of these alleles. So in this case, the numbers don't change. So P is still 0.7 and Q is 0.3. So this means that the allele frequencies have not changed. So this would be Hardy-Weinberg equilibrium. So that indicates that there's no evolution occurring. And so some of the assumptions that we have here with the Hardy-Weinberg equilibrium, again, no evolution. So we have random mating, meaning that, again, it's 100% random as far as which offspring come together or which mating pairs occur. There's no natural selection, so there's no driving force to prefer one genotype or phenotype to the other. There's no genetic drift or genetic flow or gene flow. We'll be learning more about these in a minute. And there's no evolution. So again, the idea behind Hardy-Weinberg is that from generation to generation, the allele frequencies would remain relatively the same. Okay, so then how do we know when there is evolution occurring? And the magic number for evolution to occur is 10%. So let me just sadly write this in here. So a 10% change, an increase or a decrease in those allele frequencies. So if there's a 10% change, then that means that we do not have Hardy-Weinberg equilibrium and that evolution is occurring. Okay, so if we would say, you know, originally our allele frequencies was 0.7 and 0.3, and then something happened to the population, and now the allele frequency is, let's say, 0.89 for the dominant and 0.15 for the recessive trait. Now that's above 10%, so we would indicate that evolution is occurring within that population. Or if it went down to where it was now 50-50, dominant and recessive, that would be a 10% change from this original number, again indicating there has been some type of change to the population and evolution has occurred. So 10% is that magic number. I'm not going to play it here in the recording, but the link to this YouTube, it's one of the Amoeba sisters going through the Hardy-Weinberg, just to give you a little more visual understanding about what's happening and how this principle was established. So then normally we would be doing our group exercise. Of course, we won't be doing that since we're not in class. But because there will be questions similar to this on your test, I wanted to give you some practice. So it's using both of the equations. So knowing P plus Q equals 1. So in the first example, you're told that the recessive allele, in this case B, occurs at a frequency of 0.8 in a population of crabs. So then it's asking you to determine the homozygous dominant genotypes. So again, you're given the allele frequency. You're determining the genotype frequency based on knowing 0.8 is your recessive frequency. So again, using those P plus Q equals 1, and then the Hardy-Weinberg P squared plus 2PQ plus Q squared equals 1. You can use those to figure out your answer. And then a slight twist on that, another way of using it. So in corn, purple kernels are dominant to yellow. So a random sampling of 100 kernels is taken from the population, and we're saying we do have equilibrium, so there's no evolution at this point. And we found that there are 9 kernels are yellow, 91 kernels are purple. So now you're determining the frequency of the yellow allele in the population. So again, it's important to understand the difference between asking what the allele frequency is, which is P or Q, and then what the genotype frequency is, which is your homozygous dominant, heterozygous, or homozygous recessive. And so you're using those numbers differently in those equations differently. So I did pull these so that you could have the answers. I did pull them from some examples that were online. So this YouTube video actually helps you work through these problems, and it has some other ones as well. Khan Academy is good. They have some examples that you can work through. But again, there'll be questions like this. So I encourage you to try to answer it on your own and then look up the answers to make sure you did it right. All right, so now what are the driving forces then of evolution? So if there's not the Hardy-Weinberg equilibrium and there is evolution within the population, what is making that happen? So essentially there are 4 driving forces. So mutations within a population, this gene flow that we'll learn more about, genetic drift, and then natural selection. So the rest of this is going through each of those 4 driving forces. So first of all, starting with selection. So we have natural selection, and there are 4 different patterns that we look at for selection. So we have directional, stabilizing, disruptive, and then balancing. And with balancing, it's staying the same, so it's really not changing. And then there's also sexual selection, and there's intersexual and intrasexual. So we'll go through these types of selections and then hit on the other driving forces. So first of all, directional selection. This changes the average value of the trait. Okay, so this is before whatever the selection was. This is our normal distribution. So this would indicate, a very low fitness. They're not able to produce good offspring versus a really high. And so most of them are kind of here in the middle. So in between that. So kind of a bell curve here with our average in the middle. However, after some type of selection event, now there's a shift in that average. So we still have a bell curve. Our curve looks the same, but now it's shifted. In this case, it's moved to the right. Okay. Also could have moved to the left. But whenever that whole curve doesn't really change shape so much, it just moves to the right or left, we call that directional selection. And an example of that is caused the average beak depth to increase. Okay, so the depth here of the beaks are really narrow, here really thick on this end. So originally this bell curve kind of the average was right around 9.5. However, after a drought, okay, so after we had this drought, the population that was left, all of them had a deeper beak size. So our bell curve is still there, but now it's shifted over to the right. So it went from about 9.5 to a little over 10 millimeters. So that indicated, again, a directional selection. We still have the average in the middle, but again, we've just moved it to the right or to the left. Then the stabilizing selection, this actually reduces the amount of variation. So before selection, again, a nice thick bell curve. So we have a lot kind of under the middle or here in the middle under our curve. However, after the selection event, again, we saw the bell curve, but it's more narrow. It's not as wide, indicating that there's not as much variety. Okay, so we've eliminated some of the outliers down here that were really low in fitness on both ends of this. So instead of directional, where low fitness was at the bottom and high fitness was at the top, in this case, the ends of our bell curve are both not very fit, and only the highest fit, they would be in the middle. All right, so after an event, a stabilizing event, then we have this reduced variation. An example here of this, we can look at birth weight. So for example, again, we have very low birth weights down here, and we also have very high birth weights at the other end. Okay, both of those lead typically to not very viable offspring. So of course, if the baby's really, really tiny, its chance of survival is not as good. But also the other is true. So if they're really, really big, they also have a lower chance of survival. So those that are most fit are right in the middle. So around 7 pounds or so is where we get that. So here we see, again, our bell curve that's here indicating our average birth weight. But then we also see the mortality. So again, low birth weight indicates more mortality, but then we also start to see a higher mortality as the birth weight gets too high. So at this case, we have low fitness on both ends of our bell curve, and the highest fit are the ones in the middle. So we call this a stabilizing selection. So there's not as much variation. Most babies are born at or around 6 to 8 pounds. We don't have a lot on the other ends of the spectrum. All right, then disruptive selection, this increases the amount. So stabilizing decreases variation, disruptive increases the amount of variation. So in this case, before the selection, we have the opposite. So the most fit are the ones at the ends of our graph. The ones in the middle actually are not very fit. So after some type of selection event, now actually the two at the end. So we get, instead of a bell curve, we have a curve with two peaks. And then in the middle, there's a dip where those that were not fit would occur. So again, now we're getting more variation. At the beginning, everyone was kind of the same here in this area, but now we have this population, a large population on this end, as well as a larger population over here. And an example of this, so in whitefish with relatively low or high numbers of what are called gill rakers, which are shown down here, they're the most likely to survive and reproduce. So over time, with selection, again, survival of the fittest, we get a high population of those with a low number of gill rakers and a larger population with a high number. But those kind of in the middle and between the high and the low, they're less able to survive. So then just a little graph here to kind of put this together, kind of test our knowledge here a little bit. So again, if we look at the first one, here was our before selection is at the dotted line, and then it's the same kind of shape. It's just shifted, in this case, to the right. So hopefully you figured out this would be our directional selection, so it favors one extreme phenotype to another. And then in this case, our original shape was wider. After the selection event, it's narrowed, so we decrease, so it favors the phenotype near the middle of the range. So that's the stabilizing selection. Here where we have two peaks instead of just one that was originally there. Now it favors the extreme phenotypes at both ends. That's the disruptive. And then the one that we didn't see here where there's essentially no single phenotype that's favored, that's called balancing selection. All right, now if we look at sexual selection, so we have intrasexual and intersexual selection. And the difference is that with intrasexual, we have competition between same-sex individuals. So for example, two males that are competing for a female. So for example, males fight with each other to chase them away. So again, two lions fighting for the opportunity to breed with the female. Whereas intersexual, this is between different, so two different sexes. So where the female is choosing over, is choosing a male, is typically how that happens. But here the male shows special traits or behaviors to attract the female. So we're attracting the male to the female, but males aren't competing with each other. They're trying to get the attention of the female. So that's intersexual. Another driving force of evolution is what's called genetic drift. And again, there's a video here. We're not going to click on it for this, but I highly encourage you to view it to better, understand genetic drift. So essentially, genetic drift is a random event that selects for one trait or one genotype over another. And two examples of this, so the founder effect. So if we have immigration happening, so for example, green iguanas founded a new island population. So there was a population of iguanas, and in place one, there was a lot of variation. So we had, again, our homozygous dominant, which are red, our heterozygous, which are purple, and then our homozygous recessive, kind of in a typical, 75% have the dominant trait and 25% have the recessive. So that was in the original place. However, if immigration happens, and within this population, and for example, here we have two homozygous dominant individuals and one heterozygous that are moving to a new location. If that's the original source, at this point, we have rather eliminated the homozygous recessive. So in place two, that allele frequency is going to be much different. The genotypes, these are all going to pretty much be show the dominant trait. Now, eventually we could get that recessive trait to reappear over time since we have this heterozygous. But again, what happens with this immigration and this founder effect is that we get a much smaller variety in a gene pool. So this is called genetic drift. So if we think about, again, even with people and with some populations, so for example, the Amish, for example, mainly stay together. There's not a lot of variation within their gene pool because they have a rather small community. So they migrated to an area, again, that founder effect. So only those alleles that are common in those individuals are going to be in the gene pool because they don't have a lot of variety coming in to that pool. All right, now a genetic bottleneck is a little bit different. In this case, there's some type of random event that alters the population. So we're not migrating, we're not moving to a new location with a limited number of alleles. Instead, in this case, what is happening is an event such as, like a tsunami or a hurricane or a flood or something like that has struck a population, and it leads to this bottleneck where there's a high mortality that strikes individuals, but it's a random event. So again, we're changing our gene pool here. So this bottleneck is likely to have different frequencies than what our original population has. And an example of this is what's called achromatopsia, which is vision loss that's common in the Pigalap atoll. So because multiple people in this population have this, then they continue passing it on. So this is referred to as a genetic bottleneck. So again, to where we're getting just a few decreasing that, but again, the idea is that it's random. Another example again would be a hurricane or something wiping out a large number of the population where only a few survive to repopulate. Right, then our next driver of evolution is gene flow. So this is a bit different than genetic drift. Again, genetic drift is kind of a random event that significantly changes the allele frequency. With gene flow, what we have is a more mix. We're getting more variety being added to our population. So if we show kind of the extremes here, so at the beginning we have population 1 that is all dominant. We have population 2 that is all recessive. But over time, as there's mixing of these population and of these alleles, we're getting this gene flow. So our homozygous dominant are moving into our recessive pool or population, and our recessives are flowing into our dominant population. So over time, as we're adding, so again, if we think about, individuals, especially humans, as travel has increased and now we're moving all over the globe, we're getting this gene flow of people where people in America are moving, to Africa, and people in Europe are going, to South America. So we're getting this more mixture of genes coming into a population. So it's leading to a lot more variety within our populations. And then finally, one that we've already talked a good bit about is mutations. Again, mutations are a random event that occur, but that also changes the allele frequency. So we've covered mutations. Again, mutations are just changes to the A's, T's, C's, and G's. That whenever we change one of those bases, then it can change the phenotype, it can change the protein being made, and then there can be a selection event that would select for that mutation, or that mutation may not be viable, so then it is destroyed and selected against, or it might provide an advantage and it's selected for. So then again, over time, that mutation is either increased or decreased in a population. Bacteria do this a lot. They have ways of providing different genetic information to other bacteria through a process called transformation, where they're taking naked DNA into the bacterial cell. Some of them can actually undergo almost like a sexual act, not exactly, though there's not egg or sperm, there's only the cells, but they make physical contact with each other. And then share genetic information through this process of conjugation. And then sometimes even viruses that infect bacteria can add genes from one bacteria to another. So again, there are multiple ways, especially in bacteria, to gain new genetic information. And of course, there's also chromosomal. So these are more point mutations to the left, and these are chromosomal changes. And again, we've talked about these before. So deleting parts of the chromosome, duplicating it, switching it up and down, moving it to a new location. So that's all going to change the genes. So it changes our alleles again, and then with selection, we're increasing or decreasing that allele frequency. So then if we, again, our summary slide here of these different driving forces. So here we have certain alleles are favored and increase in frequency, while others are disfavored and decrease in frequency. So this is natural selection. So again, we're selecting for an event, and then we have that disruptive, we have stabilizing. So we have different types of natural selection, but in this case, something is favored over another. Then random changes in the allele frequencies that are really important in small populations. This is that genetic drift. So again, like bottleneck or founder effect. So we're taking random, again, chances, but especially in a small population, again, people or organisms moving to a new place to reestablish or to set up a new population, or an event that wipes out a large amount of the population. Then the movement of alleles between populations, that's our gene flow to where things are coming in, which can increase that genetic variation. And then the random production of those new alleles is the mutation. So again, the key to all of this, that it is random events that lead to these changes in our allele frequencies. So again, just summarizing those driving forces, mutations are what generate new alleles in our population. And then that gene flow can increase variation by moving those alleles to new populations. Genetic drifts, we have some type of chance event that decreases the number or the amount of alleles in a population at a random occurrence. Or we also have our natural selection, where there's some type of pressure that selects one allele over the other.